So today's tutorial will be a revision of the last tutorial we did, because I know we covered quite a lot of concepts and new terms in the last tutorial. So really, I think the main aim for today is to make sure that we understand those terms. But also I'm going to extend it a little bit more by talking more about pooling estimates and meta-analysis, how we can combine the results of several different SNPs into an overall MOR causal estimate. And then also, just in preparation for Journal Club on Friday, I've put together some points and some questions that it's useful to think about in advance of that session. So just as a revision, I was going to ask a few questions firstly about our understanding of some of the concepts from last week. So we discussed a lot about observational studies and their limitations, namely the risk of confounding and also reverse causality complicating associations. So would anyone be able to explain what the concept of confounding is? As you said, you have the independent and the dependent variables. So I'd like to think in terms of examples. So, we could say that as ice cream sales go up, the number of shark attacks go up. I think I used this example the last time. And, we could say that that is one causing the other. However, as you said, there's an alternative variable that is clouding the picture here. So it's associated with both the dependent and independent variable, but it's creating this spurious association. And as you said, you can adjust for known confounders. However, it's also likely that there's unmeasured and unknown confounders that could also be mediating that relationship. So this is a problem with observational studies, confounding factors, and we can control for them, but it's very difficult to control for all of them. And that's where the benefits of a randomized controlled trial come from, because the purpose of, and I guess the strength of a randomized controlled trial is that you balance the confounding variables between the intervention and the control arm. Okay. And then the other concept we came across was reverse causality. And this is where the presumed direction of association actually happens in reverse. So the example here is that cannabis use in schizophrenia, we could think that the presumed direction here is that cannabis causes schizophrenia, but actually it's possible that this could be happening in reverse, where schizophrenia might cause people to experience symptoms that they might then self-medicate with using cannabis. So there's a reverse causation where the outcome modulates the exposure. And again, that makes it very difficult to delineate what is cause and effect, because we don't know whether it's happening in the forward direction or the reverse direction. And that's where Mendelian randomization comes into play, because we can use observational data and we can use genetic variants as a proxy to study the effect of an exposure on an outcome. And because these genetic variants are distributed at meiosis independently and randomly, you get a balancing of confounding factors. And because these variants are there from before we are even born, there's no risk of reverse causality. And just another term I wanted to introduce this week that we didn't discuss previously was the concept of instrumental variable. That's just a fancy name for the genetic variant that we use as a proxy. So we call that proxy instrumental variable. So we talk about instrumental variable analysis, but that is just another name for Mendelian randomization. And again, formally, in terms of definitions, instrumental variables are variables that are associated with the risk factor, so i.e. here the exposure, but they are not related to confounding variables, and they only affect the outcome through the risk factor. Okay, and these encapsulate the assumptions of Mendelian randomization. I think this is a key slide, and was something I was trying to emphasize when we were discussing the proposals, is that we can draw a lot of similarities between a Mendelian randomization study and a randomized controlled trial, because we identify variants that proxy different levels of exposure, and then we essentially randomize people according to this biological or genetically determined risk of exposure. So if we were to study the effect of iron on cardiovascular disease, we could select people who had variants that would mean that they have high iron are variants that they would have low iron. So again, on the right-hand side of the screen here, we see that kind of the randomization effect according to the distribution of different genetic variants. And then we would follow these people up over time to see, well, how many of these people had a heart attack, for example. And because these genetic variants are randomly distributed, confounding factors are balanced between the groups. Therefore, this reduces the risk of confounding and also reverse causality. And as I said last time, the main thing here to think about is what is the exposure and what is the outcome? And that's how we kind of divide up the Mendelian randomization in terms of its structure. And more formally, we can demonstrate and we can discuss Mendelian randomization using what are called direct acyclic graphs, or DAGs, and these just basically it's a fancy name for the kind of structure of Mendelian randomization. It's a schematic diagram. And again, exposure and outcome being the key kind of divisions here. So we look at the genetic variant, and that associates with the exposure, and the association between the genetic variant and the exposure is obtained from genome-wide association study commonly, and that's known as beta GX, and that's the association between the genetic variant and the exposure. Then we also calculate we also obtain from a secondary genome-wide association study the association between the genetic variant and the outcome. And then using the Wald ratio, as we discussed last session, we can calculate the MOR causal estimate, and that's in orange here, and that's the association between the exposure and the outcome. So that's how we set up a Mendelian randomization study. And there's three key assumptions for an MOR study. Assumption one is that there's a strong association between the genetic variant G and the exposure X, so that's a strong beta GX. And that can be calculated. It's one of the only assumptions that we can actually get empirical evidence for. And that's using, for example, the R squared or the F statistic. And as a revision, the R squared is the proportion of variability in the exposure that is explained by the instrumental variables. We can also look at the F statistic, which again measures the strength of that association. Assumption number two is that the genetic proxy is not associated with confounding variables. So again, you see the red dotted arrow. That would be an issue with an MOR study if the genetic variant was associated with a confounding variable. So that would breach the assumption, and the MOR study would no longer be valid. And the final assumption is related to pleiotropy, and as we discussed last day, pleiotropy just describes alternative paths between the genetic variant and the outcome. And for a valid MOR study, there could be no alternative pathway between the genetic variant G and the outcome Y, except through the exposure X. Okay, so there's no alternative pathways for that association to occur, that the only way the genetic variant G can affect Y is through X. And that's quite a critical assumption. We can do some statistical tests to investigate this as well. And again, pleiotropy refers to the concept that something can have diverse effects. And we mentioned last session about vertical pleiotropy and horizontal pleiotropy. And horizontal pleiotropy is a breach of an MOR assumption, because we can see that the locus or the variant is associated with both trait A and trait B. Okay, so there's two ways this could be impacting on the outcome, whereas with the vertical pleiotropy, trait B is a downstream effect of trait A. So actually it's not an alternative path, it's just on the causal pathway. Going back to our direct acyclical graph, again, horizontal pleiotropy is an alternative pathway for how the genetic variant G can affect Y, other than through the exposure X. And that's a breach of the MOR assumption, because we're no longer sure how the genetic variant G is influencing the outcome. Could it be through the exposure, or could it be through an alternative path? And to bring in some new information now, so how do we select the genetic variants, or as we can also call these, the instrumental variables? There's a number of different techniques that we can use, and you'll see different papers doing different methods. Firstly, these can be driven by our understanding of the biology. So we could look at known mutations that cause disease. Or alternatively, we could look at variants within or around the gene that associate with downstream cell signaling or biomarkers. For example, if you wanted to study the effect of ACE inhibitors, which are an antihypertensive medication, you could look at variants within that gene, the ACE gene, and how they then associate with systolic blood pressure, for example. And then we would know that those variants would act as a proxy for the ACE inhibitor drug. There's also alternative ways we can look at it. We can look at it from a more statistical perspective. We can find variants that are independently and significantly associated with an exposure at genome-wide significance. I'll discuss that in a little more detail later. And then finally, we can also use previously published variants from what other studies have used. And this might help with comparisons, for example, but fundamentally, most studies would look at biologically driven or statistically driven approaches. So from a statistical point of view, we need to select genetic variants that are independent and strongly associated with the exposure. In terms of that strength of the association and the significance of that, it needs to be usually at genome-wide significance levels. So that's p is less than 5 × 10⁻⁸, as we discussed last time. This is, again, because of the multiple testing that is going on in a genome-wide association study. We need to have more stringent thresholds to select what is a significant finding. And what does independent mean? So when we think about independence, we can think of what is an independent country, and that's something that is not affected by any other variant. It mediates an effect by itself, regardless of the presence and absence of other variants. So really, it stands alone by itself. And however, sometimes variants aren't independent. They can be what we call linked. They can happen and they can co-occur at a level greater than what we would expect by chance alone, i.e. they're not occurring at random. And importantly, these are not independent variants. Then if these variants are linked, they demonstrate what we call linkage disequilibrium, okay? And that means that they're correlated, they're related, they're almost like twins, essentially. And we need to remove these variants that are in linkage disequilibrium. We need to remove one of them, as the inclusion of these variants that are in linkage disequilibrium can introduce confounding, because we're actually not quite sure which one is mediating the effect. For example, there could be confounding because the genetic variant that is in linkage disequilibrium with the genetic variant in question could be responsible for an alternative pathway that could be affecting the outcome. So we need to make sure that the genetic variants that we include as our proxies are independent. And we can do this using computer software that removes correlated variants and selects variants that are independent. So again, we can use different thresholds for what we would define as an independent genetic variant. And again, we can use like the correlation between different variants and the correlation coefficient and set a threshold. And thresholds that don't meet this and variants that meet this threshold can then be removed. So commonly that could be the R² is less than 0.1, which means that we would remove variants that are correlated more than 0.1. I appreciate this is a lot of information, so I thought I'd put this together in an example. And this is from a paper I recently did with Depender. It's looking at the effect of interleukin-6 signaling inhibition and renal function. And I just took this out of the methodology section, and it kind of really brings to light, I think, some of the concepts we mentioned in the last slide. So I'll just read it out. So genetic variants for down-regulated IL-6 signaling were selected as independent. So again, the correlation, so variants that were correlated greater than 0.1 were removed. Single nucleotide polymorphisms, which is the most common cause of genetic variants. And these variants that were within or around the IL-6 receptor gene, and in brackets, we see the kind of position of the IL-6 receptor gene within the human genome. And we selected variants within or around that gene that then had an association with CRP in the UK Biobank. So that was a genome-wide association study. And these were at genome-wide significance level. And the reason for choosing the CRP as the downstream marker is that this is a reliable marker for IL-6 signaling, and thus variants within the IL-6 gene, IL-6 gene that associate with CRP would represent proxies for IL-6 signaling modulation. So again, in summary, we're selecting variants within the IL-6 gene or around it that are associated with downstream markers of IL-6 signaling. And because we know that these are independent and significant associations, we can therefore say that those variants would represent proxies for IL-6 signaling inhibition. So that is a more statistical approach to selecting variants, but also just as a reminder, we can use biologically driven approaches as well. And mentioned last day as well about the different forms of Mendelian randomization. And if we have access to individual level data, so that means we have the actual genetic code for all, a number of individuals, we can use different statistical techniques and different analytical techniques. But we will not really be covering this methodology because there can be issues with data privacy, whereas actually the data we will be using is publicly available summary level data from genome-wide association studies. And this is known, what we will be doing is a 2-sample Mendelian randomization study. This means that the data for the genetic associations with the exposure come from a separate genome-wide association study from the study that provides the data for the association with the outcomes. And again, this increases the power of the study as well. So in summary, methodologies that we will be focusing on in this project would be a 2-sample MOR using summary data from publicly available genome-wide association studies. And obviously we can be using many different SNPs because there can oftentimes be a number of different SNPs that would meet our criteria for what we're defining as a genetic proxy for our exposure. So this again is taken from the study I recently did looking at IL-6 signaling inhibition on the risk of chronic kidney disease. And we can see that actually use something like 20 different SNPs because all of these SNPs met the predefined criteria for our genetic proxies. And how do we approach a situation where we're getting different association, different MOR causal estimates for different SNPs? And again, these were all calculated using the Wald ratio that we discussed the last day. So what we need to do is we need to meta-analyze these SNPs. We need to combine them into an overall estimate. And this draws on techniques known as meta-analysis. And we see this in many different areas of science. And we see this there's a whole field of medical research in meta-analysis, and that is the statistical analysis that combines the results of various different trials to find a more precise estimate. So thinking about different randomized controlled trials, for example, we can combine these together if they're, addressing similar research questions and have similar methods, and we can combine them to form a more precise estimate. But similarly, in Mendelian randomization, we can combine the results of several different SNPs to form a more precise estimate. This again increases the power of the methodology. So taking an example from randomized controlled trials, it's quite a famous meta-analysis that looked at the use of antenatal corticosteroids in women who are at risk of preterm birth. And the outcome in question here was neonatal outcomes. I think the outcome was necrotizing enterocolitis. Again, quite a serious complication in preterm babies. And as we can see on the left-hand side, there's a number of different studies that were done. And these were randomized controlled trials that randomized women to receive corticosteroids or a control, and then looking at the risk ratio for the neonatal outcome. And we see that actually there's no study that had a significant finding. Okay, and that's because the confidence intervals all straddle unity. So we cannot actually say whether corticosteroids are protective or whether they have a negative effect. Okay, but once we pooled these results together, and we see the diamond at the bottom is the pooled estimate, so this combines the result of all the previous studies, we can see, well, actually, it shows that there is a risk reduction associated with antenatal corticosteroids, and that is protective against these neonatal outcomes. And that's quite interesting. So individual studies, because they were smaller, they might contain less women included in their study population. However, once we combined all these together, we increased the power, and we find that there was a significant effect. So that is really the benefit of meta-analysis, that it's providing more precise results because we're including more. In the case of randomized controlled trials, we're including more women or more participants within our analysis. And how do we calculate these pooled estimates? So how do we combine the results in the case of Mendelian randomization, these different Wald ratios for each individual SNP that we're including in our analysis? And we do this primarily through the inverse variance weighted approach. And this calculates a weighted mean for all the individuals. And that produces an estimate with a narrower confidence interval. Okay, so the relative contribution of each SNP to the final pooled estimate is inversely related to its standard error. I'll just explain that a little bit more. So as we can remember from the last day, the standard error is a measure of precision. So a more precise result has a smaller standard error, okay? And because of this, the argument is that SNPs which have a smaller standard error, i.e. they're more precise, should contribute more to the final estimate compared to a more imprecise estimate. So we are weighting the contribution of each SNP to the pooled estimate according to its precision. And for example, here on the diagram at the bottom, we can see that normally we don't weight means. We just add up the numbers and divide by the number of contributing variables. So they're not weighted. So it doesn't matter actually whether one is imprecise and one is precise. We just calculate the average. However, for inverse variance weighting, we take into account the precision and how precise that value is according to its standard error. And we weigh them. So value 1 in this instance is the most precise, so it's given the most weight in contributing to the mean. And value 2 is the second most precise, so it's given slightly less weighting. And then value 3 is the most imprecise, so it's given only it contributes only a small bit to the overall mean. So kind of in conclusion, the relative contribution of a value to the mean is scaled according to its precision. And we can see that here in the forest plot for the IL-6 paper. We can see if we look at the last value in red, so inverse variance weighted, this one, we can, sorry. We can see that it's taking into account all of these different SNPs, and actually the confidence interval is negligible. Essentially, it's very small because it's taking into consideration all of these SNPs. And again, I would predict that this SNP here is providing the most contribution to that value because it has the smallest confidence interval. Again, the confidence interval is the bar either side of the circle. That's a 95% confidence interval, representing it's a range of values with an associated 95% probability that the true estimate would be contained within that range. So how do we formally calculate this? We'll go through the calculations in a little while, but the weighting we use is 1 over the standard error squared. And then to calculate the inverse variance weighting, we sum up the Wald ratio multiplied by its individual weighting, and then over the total of the weights. Okay, I think sometimes it's easier to see this in practice to understand it, but again, the concept here is that we are weighting according to the standard error. So if the standard error is large, 1 over a large number would be very, very small. So actually that value then will be weighted, given very little weight. And then if the standard error is very small, that's going to be a large weighting then associated with that Wald ratio. So that's how we are that's how we are scaling the values according to the standard error. I'm just taking this panel from Depender's work on iron status on coronary artery disease. So we see that there's 4 different metrics of iron exposure here. So serum iron up in the top left. On the top right, we have transferrin saturation. On the bottom left, we have ferritin. And on the bottom right, we have transferrin. And again, these are all the genetic association, the MOR causal estimates for the effect of iron on coronary artery disease. And we have 3 different SNPs. So let's discuss the effect of iron on coronary artery disease. So the panel, the figure up on the top left, we can see that SNP or S1800562 is associated with a decreased risk of coronary artery disease. And the confidence interval between 0.79 and 0.99, so that shows that it's significant reduction. The next SNP down, so ORS 1799945, the 95% confidence interval straddles unity, so it's a non-significant finding. And similarly, ORS 855791, despite the fact that the odds ratio is 93% or 0.93, so a 7% reduction in the odds of coronary artery disease, the confidence interval straddles unity, so it's a non-significant finding, because actually, if we look at the confidence intervals, it could be anywhere from a 15% reduction in the risk of coronary artery disease to a 1% increase. However, we then calculate a pooled estimate. So again, the diamond denoting a pooled estimate. It's a form of meta-analysis, and this was done using the inverse variance weighting approach that we previously discussed. And we can see that once we pool these results together, we find that actually the overall odds ratio is 0.94, and the confidence interval is 0.88 to 1. So again, it's not really straddling unity. So it goes from it could be anywhere from a 12% reduction to no effect, essentially. And that is how we interpret the forest plots and interpret the odds ratios. Again, it's also important to note that we have to remember binary outcomes, so coronary artery disease, yes or no, need to be exponentiated at the end to convert these to an odds ratio. So final revision before we go into some of the calculations. This was taken from the OR package, 2-sample MOR package. And again, the objective here in a Mendelian randomization study is to infer the causal effect of an exposure on an outcome. So we take SNPs, which are genetic proxies for an exposure, and then we study the effect of those on outcomes. This is assuming that the assumptions of Mendelian randomization hold. This should be independent of the effect of any confounders, and reverse causality shouldn't be an issue. We select the variants that would represent the exposure. So commonly these could be known as instrumental variables. We can use several different ways of doing that. We can use it using biological knowledge, statistical analysis, and we get this data from the summary statistics of genome-wide association studies. And then we also get the data for the outcome, and we find the beta GY, so that's the association between the genetic variance and the outcome. And then we also need to harmonize these effects. So harmonization refers to the fact that the effect allele needs to be the same for the exposure and the outcome associations. If they aren't, we can harmonize these. So that means we can reverse the direction and say which allele is the effect allele and which allele is the non-effect allele by changing the sign of the effect. I'll go through that in more detail when we actually do the calculations. When we do these in packages and OR, it happens automatically, but I think it's important to realize that we need to harmonize to make sure that the effect allele is matched between the outcome, or between the exposure and the outcome data. So before we go to the calculations, I just wanted to give some advice for the journal club presentation. So appreciate this might be a new experience for us. Firstly, I think it's important to just describe an overview of why the study was done, why is this important, and then also look at why Mendelian randomization was a suitable methodology to answer this research question. For example, has there been no randomized controlled trial data or randomized controlled trial study being done? Is the RCT data conflicting? Is it not possible to do this form of study using other methods, for example? And then what type of MR study was this? Was this using a two-sample approach, using summary data from publicly available genome-wide association studies, for example? How do they select the genetic variants? Where do they obtain the outcome data from? And what methods were used to calculate the MR estimate and the pooled estimates? And also then I think it's important to look at the assumptions of the Mendelian randomization approach. So for example, is there any possible breaches of these assumptions? Have they discussed pleiotropy? Have they done any analysis to look at pleiotropy? Sensitivity analysis is where we change some of the assumptions that we make about our study and assess whether that has an impact on the results of our study. So that's another way that we can kind of test how robust our methods are. And then finally, what are the conclusions of the study? So for a journal club, typical format would be about 15-20 minutes of a presentation and then 10 to 15 minutes of questions. Again, just following kind of and addressing the questions that I put on the slide here. But again, it just goes through kind of step by step how to do the analysis. So as I said, step one would be to harmonize the effect allele for the exposure and the outcome. And if there's a discrepancy, we can flip the direction of either the exposure or the outcome so that they align. So here is an example that I've taken from the internet. So there's 3 different SNPs, right? So RS 1, 2, 3, 4, 5, 6, 2, 3, 4, 5, 6, 7, 3, 4, 5, 6, 7, 8. The effect allele is AGG. Okay, and this is, you know, that's the replacement. So that's the variant. Usually people might have G, but in this case, the person would have a different nucleotide base. So they have an A at that position. Here they have a G instead of a T. Here they have a G instead of a C. Okay, but actually you can see here when they look at the outcome data from the genome-wide association study, you see that there's different effect alleles. It's actually the effect allele here is G, or no, the effect allele here is T, the effect allele here is G. Okay, but we can reverse these directions by multiplying the effect estimate by -1. So we're changing the sign, and that means that the effect allele or the other allele becomes the effect allele. So G becomes the effect allele by multiplying the effect by -1. So we flip the direction of the SNP by reversing the sign of the beta estimate. Does that make sense? And then after that, we found the Wald ratio. So that's the beta for the outcome over the beta exposure. And again, if it's a binary outcome at the end, we need to exponentiate that to find the odds ratio. The standard error is found using this calculation. And then we use a standard error to calculate the 95% confidence interval. So the upper limit is the estimate plus 1.96 times the standard error. The lower limit is estimate minus 1.96 by the standard error. But again, at the end, if we need to convert this to an odds ratio, we multiply, or we exponentiate after we have added on or subtracted the standard error. Then we can pool several different SNPs together to form a more combined estimate, and that's using the inverse variance weighting approach. There are other approaches, but primarily we'll be using this one. And again, the weighting that each SNP is given is according to 1 over the square root of a standard error. And then we can calculate the standard error for the inverse variance weighted estimate using this formula. Okay. So if it's okay, I'll go through the calculations with you now. So this is based on Depender's work that he published in 2017, look at the effect of iron on coronary artery disease. So again, I've taken just 3 of the metrics, so serum iron, transferrin saturation, and transferrin itself, and we can see the effect allele, the non-effect allele, effect allele frequency, and then the F statistic, the R squared statistic, and then GX, so that is the association between the genetic variant and the exposure. So here the exposure is iron, and the standard error of that GX, and then the p-value. Okay, so F statistic here is mostly greater than 10. Actually, it's all greater than 10. So there's strong, what we call strong instrumental variables. So that MR assumption 1, where the strength of the outcome, the strength between the genetic variant and the exposure is strong. Now, these are only attribute, these only contribute a small amount to actual variability in iron. So 1.3% for the RS1800562 SNP. So 1.3% of the variability in iron is explained by that SNP. So not really contributing a lot, but again, it's significant. So it's 2.72 by 10 to the minus 97. So it's highly significant. Okay, then we have these SNPs again. So remember these SNPs, they're the codes for the SNPs. We have these SNPs again, but we're looking at the outcome data. So this was from genome-wide association, I think it's cardiograms it was taken from, and that had 34,997 cases and 49,512 controls. So about 35,000 of these people had coronary artery disease. So I think that was defined using heart attacks, for example. And about 50,000 people didn't. And then they did a genome-wide association study where they looked through the association between different variants and the risk of coronary artery disease. And they found that these SNPs, that was the association. Okay, and how do we interpret this GY? So that's the association between the variant and the outcome. So I could expatiate that. So that is 0.9598, so about 0.96. So that's a 4% reduction in the odds of coronary artery disease for every copy of this allele that a person has. So if you have the nucleotide A at that position, that's if you have this SNP, you have a 4% a reduction in the odds of coronary artery disease if you have for every copy of that SNP that you have. We set up the MR study. So again, the SNP, the effect allele, the non-effect allele, and this is and again, this is for a transferrin SATs. So this is the association between the genetic variant and the exposure. Remember 0.577, and that is from here. Okay. That's a standard error of that association. And then we have the outcome data. So that's GY, and that is taken from here. So again, these are the exposure and the outcome data are taken from two separate non-overlapping genome-wide association studies. And then we calculate the Wald ratio for each of these SNPs. So that is the outcome data. So this divided by this, and that gives you that value. And then we can calculate the Wald ratio. The standard error is calculated using the standard error of the outcome association over the beta for the outcome. Okay. And then we can calculate the odds ratio by taking the exponential of the Wald ratio. And then we calculate the 95% confidence interval by taking the Wald ratio plus 1.96 times that. And so that gives us the upper interval. And then to calculate the lower interval, we take the Wald ratio minus 1.96 times the standard error. And then at the end, we exponentiate these. So here, this is the calculation I'm using. So I'm taking H2, which is the Wald ratio. I'm subtracting 1.96 times that standard error. And then at the end, I'm exponentiating it to find the odds ratio. And that gives us the odds ratio. Okay, and that is the calculation. So remember these figures for this. So 0.93, 0.93, 1.03, 1.03, 0.93, 0.93. Now I'll just explain the inverse variance weighting approach, because I think that's new information for this week. So here we have all the SNPs again, the Wald ratio, and the standard errors. That's just taken from up here and placed down there. Then we calculate the weights. Okay, so again, the weighting is, the higher the weighting, the lower the standard error. So you can see that this standard error is the lowest, so it's given the highest weighting. This standard error is the middle, or is the greatest, so it's the most imprecise estimate, so it's given the lowest weighting. And this one is the middle one, so it's given the middle weighting. So that's 1 over the standard error squared. And again, how is that actually occurring? Is because 1 over a really large number is a very small number. Okay, and then 1 over a small number is a very large weight. Okay, so then if we scale the Wald ratio according to its weight, so that's just scaling means multiplying, so the Wald ratio is multiplied by its weight, so that is this number multiplied by that number gives you that. Okay, and again, we calculate that weight, the weighted Wald ratio for all those variables. And then to calculate the inverse variance weighted estimate, that is the sum of the weighted values over the sum of the weights, and that gives us this number. And then at the end, we exponentiate that to form the odds ratio for the inverse variance weighted estimate. The standard error is the square root of 1 over the sum of the weights. So again, we calculate that. And then to get this value, that is the inverse variance weighted plus, or sorry, for the upper limit, for the lower limit, it's the inverse variance weighted estimate minus 1.96 times this value, then at the end exponentiated to form that odds ratio. And for this value, that should read the upper limit, sorry. That is the inverse variance weighted estimate plus 1.96 times the standard error, and then at the end exponentiated, and that gives us that value. Okay, so actually what we're seeing here is that the odds ratio for the pooled estimate is 0.94, with the confidence interval being 0.91 to 0.99. And that's for transfer and SATs. And if we go back here, sorry, that should be rounded up. So 0.95, 0.95 with the range going from 0.91 to 0.99. Correct. Okay, and then the diamond reflects the confidence interval. The dotted line is the inverse variance weighted pooled estimate. And that's how you do it.